Talk about the first section of chapter 20, which are talking about evolution of populations. So this is going to be really important because up until this point we were just talking about evolution in general. And in section 20.1, 20.2, we really get to how does evolution really happen? What really drives evolution? And how can we tell if it's happening? And we're going to focus on quite a bit practicing the Hardy-Weinberg equilibrium equation, which is an equation we use to tell if evolution is happening or not. If we're looking at a species over time and how we can quantitatively or mathematically tell if evolution is happening. So, first of all, on a small scale, scientists look at populations to measure whether evolution is happening. Like in the video we watched with the grants and the medium ground finches, that population of medium ground finches they saw evolved, their beak depth changed. What's important to remember is individual organisms don't evolve. Populations do. So, I'm an individual organism. I can't evolve on my own. The whole human population can and has evolved over time. A population is just a group of organisms of the same species that interbreed with each other that live in the same place. So just a group of humans or finches that are the same species that live in the same place and they're breeding with each other or creating offspring with each other. And when we refer to populations evolving on the small scale, we think about microevolution. So this isn't new species happening or things like that. It's just that individual alleles, or the frequency of an individual allele in the population, is changing over time. So what I mean is that in a given population, say of finches, if, let's say, a deep beak gene exists, and it can be represented by dominant allele A, and a shallow beak depth in the finch population is recessive A. Before the drought, maybe the frequency or the prevalence of this dominant allele was only 46%. That means only 46% of the birds had a copy of the A allele. And the frequency of the recessive A allele was 54%. So, out of the total number of alleles in the population, 46% were capital A's and 54% were small a's. After the drought, maybe the dominant A was up to 60%, and the recessive or shallow allele was 40%. In this situation, evolution has happened. Microevolution has happened. There's been a change in allele frequency. Here at the top, out of all the alleles in the population, 46 of them were a dominant A, deep beak allele. After the drought, now that has increased, 60% of the alleles are the deep beak allele for that specific gene. And this change has happened because of natural selection. The deeper beaks can crack those larger nuts a lot easier. So this is microevolution. The frequency of a given allele is changing over time. And natural selection happens to be what is causing this change. Right. So, needless to say, like we talked about with the finches, genetic variation is what makes evolution possible. So variation of alleles makes evolution possible. So if all the birds had the same alleles let's say every bird had 2 copies of the dominant A allele. Evolution couldn't happen. Alright? You need to have both the possibility, some birds carrying genes for different size beaks, or the beak depth to be a different size for evolution to happen. So very important. Genetic variation is what makes evolution possible. Also, the greater amount of variation is, there's more potential for evolution to happen. The more different alleles there's out there, the more natural selection has to work with. And there's 2 ways we can measure genetic variation. It can be measured by the average percentage of heterozygous at each locus in a population. So for example, if we're looking at the beak depth locus from the last slide with A deep and recessive A shallow, we can measure genetic variation by saying at this locus 34% of the birds are heterozygotes. So one measure of genetic variation is that an average number of the population that's heterozygote. Another thing we can measure is the nucleotide variation. So how many differences, what are the amount of differences in the population of a sequence of DNA? So we have DNA strand 1, DNA strand 2, and we look for differences, much like we did with amino acids. This isn't always, this isn't always very reliable, because those differences could be maybe in introns, where they don't even matter. They could, they get spliced out. Maybe those variations are silent. They are a point mutation that codes for the same amino acid, doesn't change the phenotype. Or maybe they're just in a regulator when it doesn't affect the expression of that gene at all. Alright? But the most important thing is that genetic variation is the source, the fuel for evolution. The sources of genetic variation are numerous. One is mutation. The main source of genetic variation at the gene level is mutations. So, for example, duplications, when different genes get copied over. Point mutations, where we have like an A gets changed to a C, or something like that. Deletions, inversions, those are all examples of mutations. And mutations are just basically a shot in the dark, because they aren't specific. You can never, they're basically random where they end up occurring. Some can be silent, meaning that they don't actually change anything phenotypically. A lot of them are just in somatic cells, meaning not the gametes. So mutations are only passed on if they occur in the gametes. So for example, mutations that happen in all the other cells besides sperm and egg cells don't get passed on, and they don't contribute to evolution. Only mutations that happen in the gametes get passed on. So like we talked about, duplication of genes is a key source of variation, because for example, the protein that gene is supposed to be creating can still be created with the first copy of that gene, and the second one can mutate and form novel functions. For animals like ourselves, sexual reproduction is a giant source of genetic variation. It's the main source of variation for sexual reproducing organisms. And there's 3 ways that sexual reproduction increases genetic variation. So, 1, crossing over in meiosis, / maybe mistakes in meiosis. Independent assortment. So if we think back during the anaphase of meiosis 1 and 2, homologous chromosomes and sister chromatids can go to either side of the cell, and it really happens randomly. And then 3, random fertilization. So any one of the trillions of different sperm cells or trillions of different egg cells made can fertilize each other. And so that's just another layer of different possibilities of genetic variation. But crossing over, independent assortment, and fertilization are the main mechanisms by which reproduction increases genetic variation. All right? So without mutations and sexual reproduction, variation wouldn't occur naturally and evolution couldn't take place, especially at the population level. So how exactly do populations evolve? Well, we saw natural selection, that some alleles are beneficial, and in times where those beneficial alleles confer advantages in survival, they will increase. But for a population to evolve, the allele frequency, such as A versus A, must change over time. So in, like I said, the first generation, 40% A and 60% little a, and then second generation maybe 45% and 55%, and the third maybe is the opposite, 50-50. If you look at this, the frequencies are changing. This dominant allele is becoming much more common, prevalent, and the recessive allele is becoming much less prevalent. This really can only occur with reproductive isolation. So a population in a specific environment can't breed with other organisms or other members of that same species in different populations. So reproductive isolation is very important for evolution to happen. Right? And what's happening is the gene pool is changing. So the gene pool of a population is just all the copies of every allele for every organism in that population. So, for example, like my example down here. If we have 500 flowers, and 320 are red, so they have CR, 160 are pink, they have like a CW, CR, and 20 are white, 2 CWs. This is codominant, by the way, where the heterozygote is a mix between the red and the white. So, for example, the gene pool for this population would consist of 1,000 alleles, right? Each flower has 2 alleles, so all the flowers together have 1,000 different alleles. The prevalence of the CR allele is, well, we have for these 320 red flowers, they have 2 copies, so there are 640 R's here, plus 160 R's here. So in the total population, there are 800 copies of the CR allele. For the white allele, then, the CW, there is 20. In the white, they have 2 copies, so 20 individuals with 2 copies, that's 40. plus 160, or 200. So there's 200 copies in this population as well. That means the prevalence of the R allele is 80%, and of the white allele, or CW, equals 20%. So I did that. That's why I figured out the prevalence, or the frequency of the allele. CR is 80%, CW is 20% in this population. Alright? So, the gene pool is just all 1,000 of these alleles. For example, if there was only one allele, so maybe in this population, none of them were white or pink. They were all red. They were 100% CR. We would say that that allele is fixed. It is fixed if there's 100%. No evolution can happen if an allele is fixed. Alright. So, now we need to look at the Hardy-Weinberg equation. And we use this to test whether a population is evolving. It's an equation, and we can see if a population is evolving by comparing the allele frequencies from generation to generation. So, from generation to generation, because we know that our definition for evolution is the change of allele frequencies over time. And we're going to compare what the allele frequency should be if the population was not evolving. So if the population is not evolving, the allele frequency shouldn't change. Not evolving, the frequency is not going to change. So if there is change in allele frequency, for example, if there change from this 80 to 20 over time, the population is evolving. And we use the Hardy-Weinberg equation to do this. If a population is not evolving, allele frequencies will stay the same. Like cards in a deck, there will always be 4 aces, 4 jacks, 4 kings. That deck isn't evolving. No matter how we deal or how we shuffle it, it will always be the same. So we're going to think of this as alleles as like colored marbles in a bag. So going back to the CR and CW alleles for our flowers, we said that the red allele, or CR, was 80% frequency in that population of 1,000, and CW was 20% in population. And in the Hardy-Weinberg equation, P equals the frequency of one allele, and Q is the frequency of the second allele. Either way, you can mix and match either one, but for this example, P will be the frequency of the red allele, so 0.8, and Q is the frequency of the white allele, so 0.2. So up here I'll just put that this is going to be a CR, and Q is going to be our CW. Alright, so we think of our gene pool as like really put a giant bag or a giant pool, and we're going to pretend like we're picking these alleles out at random. Each generation we pick the alleles from the previous one out at random. So each egg cell has an 80% chance of being red and a 20% chance of being white. And each sperm cell has an 80% chance of red and a 20% chance of white. Alright? So, the first part of our Hardy-Weinberg equation is that P plus Q is always going to be equal 1. Alright? What does that mean? The frequency of the red allele plus the frequency of the white allele is always going to be 1, or 100%. That makes sense, right? The frequency of our first allele plus the frequency of our second allele is always going to be 1. So 0.8 or 80% plus 0.2 or 20% is always going to be equal to 1. Alright? Secondly, now we're going to start drawing marbles out of here. And we're going to think, too, what is the probability of getting a CR? So, if we pick our first marble out of there, there's a 0.8% chance of it being a CR. And if we pick our second marble out of there, to be a CR, it also has a 0.8% chance. So the probability of picking 2 CRs out of this population is 0.8 is our P times P, or P squared. So if a population isn't evolving, we would expect that every generation, the possibility of picking the CR genotype, or the amount of CR genotype, is the probability, or the frequency of the P allele squared. Same thing we could do with the Q. With CW times CW would be Q, which is 0.2, times Q, 0.2, Q squared. Alright? So, we have P squared plus Q squared. What about the heterozygote? So what about CRCW? CRCW. Well, that is going to be a little bit different. And I'm going to show you that example here on the next page. Alright? So, this is kind of what I was just going over. If the CR allele, the red allele, 80%, P is 0.8, white allele, CW, 20%, Q is 0.2, there is an 80% chance my sperm is going to have a CR, and there's going to be an 80% chance my egg is going to have a CR. So, the frequency of the CR phenotype is going to be P squared, or 0.8 times 0.8. Same thing with CW. That's going to be Q squared, 0.2 times 0.2. What about my CRCW? Well, my sperm could have a CR, which is 0.8, and my egg cell could have a white allele times 0.2. But I also could have my sperm cell have a white allele, and my egg cell to have a red allele. So, if 0.8 was represented by P, and 0.2 is represented by Q, P times Q plus P times Q, or 2PQ. Alright? So this is how I can use, if I know the allele frequency, like right here, I can figure out if we're in Hardy-Weinberg equilibrium, meaning no evolution. If there's no evolution, this would be my frequency of each genotype. So if we're in Hardy-Weinberg equilibrium, the number of homozygous dominants in this example would be P squared, of homozygous recessives, or 2 white alleles, Q squared, and the heterozygote would be 2 times P times Q. So we can write this as the Hardy-Weinberg equation. There's 2 equations. One we saw back here, that P plus Q equals 1. Second is P squared, the prevalence of the CR genotype, or the CR genotype, plus Q squared, the prevalence of the CW genotype, plus 2PQ, the prevalence of the CR-CW genotype, equals 1. And so if a population is in Hardy-Weinberg equilibrium, these 2 things will always occur. So, important things to remember. P equals the allele frequency. P squared, or Q squared, equals a phenotype or genotype frequency. Alright. So, there's 5 things that have to be true for a Hardy Weinberg equilibrium to be reached, for no evolution to happen. So when we say Hardy-Weinberg, so a population is in Hardy-Weinberg equilibrium, that means there's going to be no evolution. That means that these same percentages of offspring will be true every single generation. No allele frequency is going to change. No mutations can happen. Remember, that is genetic variation. 2, random mating, so no mate preferences. 3, no natural selection. No genotype or allele is preferred, or it confers no adaptive advantage. 4, it's an extremely big population. So smaller population sizes, just random fluctuations in allele frequency can cause big changes in the overall population makeup. So it has to be a very big population. And then 5, no immigration. There can't be any individuals going in and out of the population. So if any of these things are not true, the population isn't in Hardy-Weinberg equilibrium. All have to be true for Hardy-Weinberg equilibrium. Alright. The last thing I'm going to do is we're going to go through a sample question using our Hardy-Weinberg equilibrium equation. And you'll notice on your, formula sheet that Hardy-Weinberg equilibrium equation is right down here. That's what we're going to be using, okay? And so what we need to remember is P plus Q equals 1, and P squared plus Q squared plus 2PQ also equals 1. So we have a sample population in which you know that the percentage of the homozygous recessive phenotype, AA, so that's our Q squared, is 36%. Using that 36%, calculate the following. Frequency of AA genotype, frequency of the A allele, frequency of the dominant A allele. Alright, so we have the Q squared. We need to know the P squared. We need to know all of these things. If we know that this is Q squared, this is 36%. What is Q? Q is going to equal the square root of Q squared. Q squared equals 36, then Q is equal to 6. Q is equal to 6. So my Q is 6, or 0.6 actually. 0.6. If Q is 0.6 and P plus Q has to equal 1, then P has to equal 0.4. So, the frequency of the A allele would be that 0.4, or 40%. Nope, sorry. Messed up. The frequency of the recessive A allele is Q, is 0.6 or 60%. We were given that in this problem. We figured that out. The frequency of the A allele, which we found by just subtracting, is 0.4 or 40%. The frequency of the AA genotype, they just gave it to us, was 36%, or 0.36. The frequency of the double dominant AA is 0.4 times 0.4, or 0.16, or 16%. What's the frequency of the dominant A recessive heterozygote? 2 times P times Q. So 0.4 times 0.6 would be 0.24 times 2 is 0.28. So A equals 0.48. So 0.48. So now to double check, we need to see that P squared plus Q squared plus 2PQ equals 1. So P squared, we saw from right here was 0.16 plus Q squared 0.36 equals 52. 0.52 plus 0.48 is 1. We know that's right. The frequencies of the two possible phenotypes if A is completely dominant over A. Alright, so we kind of figured this out. It's just using these two equations, plugging and chugging. Alright, that's it. We'll go through this in more detail in class.